So now that we've talked about the cell shapes, arrangements, and different sizes that we might see, it's time to zoom in onto a single cell and really look at cellular structures. And I want to begin with the cell membrane. So if we look at this somewhat simplified model of the prokaryotic cell, we would point our attention to the plasma membrane, and that's where we're going to zoom in at this point. And I want to speak just about the qualities, nature, structural components of the plasma membrane. So the plasma membrane is created using a couple of things. One, a phospholipid bilayer. So one macromolecule lipids, very much important in the membrane. Also, though, proteins are a big part of comprising this. So we sometimes say that within a phospholipid bilayer there is a mosaic of proteins. And in fact, one of the ways that we refer to the cell is using the fluid mosaic model. And we'll talk about that in a moment, but it's a mosaic of proteins within a phospholipid bilayer. Whenever I draw a phospholipid bilayer, you're going to see the exterior of the cell indicated on the upward side and the interior on the downward side. And if I don't label it, that's exactly what you can assume or presume whenever you look at a picture. In fact, really any picture, you can make that assumption. Now, let's talk about the proteins and how we might differentiate certain types of proteins within this bilayer. One type of protein is the type of protein that spans the membrane entirely. That is, there is an extracellular region, an intracellular region, and there is a transmembrane segment of the protein. We sometimes call these transmembrane proteins. We also often call them integral membrane proteins because they are completely integrated into the membrane and they span it 100%, right, from one side to the other. The other type of protein, these are called peripheral membrane proteins because they're associated with the periphery of the membrane. They don't span it entirely. So they are, as this red protein shows, somehow associated with the membrane on either the intracellular or extracellular side or either the cytosolic or the exterior side. Either way, they're going to be called peripheral membrane proteins. So these characterize the mosaic of proteins within the membrane. There are some other things that we should talk about in the membrane as well. Notice this kind of weird looking orange sort of structure. Well, it turns out that membranes require some sort of molecule that helps to maintain the fluidity of the membrane. What I mean by that is that that's jargon or lingo for saying that these molecules keep the membrane from freezing when it gets too cold, and they keep it from melting when it gets too hot. Now, some of you might be thinking, now, wait a minute, isn't that what cholesterol does? Yeah, it is. But cholesterol is only found in animal cells. Now, hang on. There's a homework question that makes me inclined to say that a second time. Cholesterol is only found in animal cells. It is not found in bacteria. It is not found in archaea. It is not found in plants. So the molecule that plays the role of maintaining fluidity or modulating fluidity within the bacterial world are the hopanoids. So these stabilize the membrane. They also modulate its fluidity, keep it from melting, keep it from freezing when temperature fluctuations occur. Another molecule that I want to draw your attention to is the carbohydrate moiety that you might see attached to either a phospholipid or a protein, giving it a unique name. So here we have a lipid, a phospholipid, with a carb region attached to it. So we call it a glycolipid. The glyco part of that name indicates the carb region. So this glyco refers to the carb, and then of course we see that it is a lipid. So glycolipid. That is two macromolecules in one, right? Both carbohydrate and lipid in that molecule. Likewise, you could get a carb attached to a protein, and then I challenge you to guess what that might be called, right? We know that it would have a two-part name as well. So, shoot me an email for a little extra credit. If you listen to this on Wednesday, I will accept extra credit if you email the name of a protein that has attached to it a carbohydrate moiety. Let's talk about that fluid mosaic model. So remember that I mentioned that the membrane it's a very distinctive structure. In fact, membranes can actually be used to distinguish or identify different cells. They're very unique. That is, some cells have a higher protein composition, some have a higher lipid composition, and that ranges. But one thing that doesn't change is the behavior of the membrane. That is to say that membranes are governed by a type of fluidity. The proteins within the membrane, they can diffuse laterally, they can rotate. You guys will appreciate this. It's kind of like swing dancing. The motions that are allowed for proteins are back and forth and turning around. So back and forth and turning around, it's like swing dancing. The motions that are not allowed for proteins are complete flip-flops. So they swing dance, but they don't do gymnastics. Okay, some of you know how that goes. Maybe you're not so much on the handsprings and whatnot, but you like to swing dance. So that's true of our fluid mosaic model as well. The allowed motions are horizontal or lateral and rotation. And that is what the fluid mosaic model says. It also says that the membrane is a selectively permeable barrier with proteins interspersed like a mosaic. So it was Garth L. Nicholson and Jonathan Singer who described this model in 1972, and we now understand that it dictates the dynamic nature of proteins, or of membranes in general, of both the proteins in the membranes and the phospholipids as well, because phospholipids also can diffuse laterally and rotate, and those are the motions allowed to them. Fantastic. Let's go ahead and look at an animation. Yes, cheesy animation, be rate, but I'm tooting my horn on my animation skills because that's about as good as I can get. So let's look at an animation that sort of helps to show the fluidity of a membrane. So notice the lateral movement of the proteins. They are not flip-flopping. It's hard to show rotation with my animation skills, but they would be rotating too. The phospholipids, likewise, would also be doing that. So a membrane is dynamic, not static. It doesn't stand rigid. It likes to have a little fun swing dancing. Let's answer some questions, just a few for thought. I'll throw at you. Would the amino acids found in the membrane-spanning region of an integral membrane protein love or hate water? Okay, let me make this clearer. So say we're looking at this blue protein, and we're talking about the amino acids that are right here, maybe right in this area here. They're exposed to the very, very water-hating tails of the phospholipids. So likewise, they too would be water-hating, right? Like dissolves like. So we would find a lot of hydrophobic, water-hating amino acids in this region of the protein. Oh, hey now, to earn full extra credit on that email that everybody's going to send me on Wednesday. By the way, I won't accept it any day but Wednesday. If you send that email to me and also tell me in that email an example of at least 2 amino acids that would be found in the transmembrane region of an integral membrane protein, that's the only way you'll learn all the extra credit, right? In fact, I'll only accept it if you tell me this and the answer to the other question. I'm getting downright like, you know, getting the whip out here and making this hard. So to earn full extra credit, you've got to have both of those things. Awesome. Now, would it be easier to extract a peripheral membrane protein? Remember, peripheral membrane proteins are associated peripherally, right, like this. So this could be a peripheral membrane protein right here. Would it be easier to extract that one, or would it be easier to extract an integral membrane protein? This is a pretty easy question because think about it. An integral membrane protein, you'd have to rip it out of the bilayer in order to get it. That would require something that would solubilize the bilayer. So in order to get an integral membrane protein out, you would have to treat with detergents, you would have to solubilize the membrane, and it would be quite a task. Whereas all you'd have to really do to get a peripheral membrane protein is just to add some salt and cause some of the proteination states on the amino acids to change, and really it would be very easy to isolate a peripheral membrane protein. So answer to that question, it'd be a lot easier to isolate a peripheral membrane protein than it would be to isolate an integral membrane protein. So what are some functions of an integral membrane protein? Now this is where I think the questions get easy. Some of the things that an integral membrane protein does is transport, bringing molecules in, taking them out, right? Transport. Another thing that an integral membrane protein might do, and I bet Shannon is thinking about this even as we speak, it might be a receptor. So maybe there's some molecule that binds to it and it triggers some intracellular response. Maybe this particular transmembrane protein or integral membrane protein contacts some other protein, sends a signal, and there's some sort of cascade within the cell, this long series of events of signaling. So it might be a receptor. So there are a multiplicity of functions that an integral membrane protein might have. Another one that I want to draw your attention to is in energy generation. So sometimes integral membrane proteins actually play a role in something that we call the electron transport chain, and I'm about to get there. Some of you will appreciate very much that the membrane is kind of like a bouncer at a dance club. So, you go to a fancy dance club in Denver and remember you get in a line and all of a sudden you recognize that who's getting let in the doors preferentially. Probably the girls with low molecular weight and the ones that don't want to drink water. Hell no H2O, right? So this is very much like the membrane. Small hydrophobic molecules, molecules with low molecular weight that are water-hating and can get past those water-hating tails of the phospholipids. So small hydrophobic molecules with low molecular weight, such as gases, such as oxygen, carbon dioxide, these can pass through via simple diffusion. They follow their concentration gradient, meaning they go from areas of high concentration to areas of low concentration. Now, other molecules, of course, require transporters. So as promised, we're going to talk a little bit about the electron transport chain. Now, don't get scared, because the first time I tell you about the electron transport chain, you're going to be like, whoa, I don't get it. But then I'm going to tell you about it again, and then again, and then again, and then again, and then finally you're going to be like, man, I totes get that electron transport chain. I understand every nuance of that particular system that there is out there to understand. So I want you to bear with me as I first introduce it for the very first time. And then from here, we become more and more nuanced in our understanding of this energy transfer system. So prokaryotes, some of them have an electron transport chain, and some of the integral membrane proteins on their cellular cytoplasmic membrane are involved in the electron transport chain. But it's true that only some prokaryotes have this, not all of them do. And in fact, only those that are capable of respiration or photosynthesis have these transporters, these components of the ETC. So these components of the ETC play a very unique role. That is, they take electrons and they pass them through the integral membrane proteins and they harness the energy that comes from those electrons. And once they've harnessed that energy, then they can use it and convert it to a different form. They convert it into a proton gradient. So they use the energy from the electrons to pump out protons and to make a gradient of those protons. So outside of the cell, there becomes a much higher concentration of protons. In fact, they're in such high concentration that it's like a pent-up battery-like force that can be tapped into. Now, for those of you engineers, you're going to recognize that this is very, very similar to the kinds of energy that we get by having, say, a forebay that is upstream at higher level than above a dam that can be tapped into to turn a turbine, right? And that turbine can be converted from mechanical energy into electrical energy, and many people can get their energy from a turbine that's turned from water energy. This is very similar, right? A proton gradient is a lot like that pent-up water that's at higher ground that can turn the turbine. These protons are pent-up force that can rush into the cell, and they can turn a machine that makes ATP. So this is a very similar sort of process. So this electrochemical gradient can be tapped into to make energy. So one of the functions of a proton motive force, or a PMF, is the synthesis of ATP. It drives that synthesis. So the PMF drives the synthesis of ATP. But another thing that this pent-up battery-like proton motive force can do is when it rushes into the cell, it can also transport other molecules. hopefully everybody's seen Finding Nemo. And remember in Finding Nemo, you remember the turtles, and they're like riding the EAC, dude, right? They're all kinds of taking advantage of the Eastern Australian current. Well, this is very much like the Eastern Australian current. That is, the EAC is a lot like the PMF, right? And molecules can ride the PMF to get into the cell. So another thing that this proton motive force can do is it can be tapped into for transport. Lastly, it can be tapped into to rotate flagella. As these protons swoosh back into a cell, it can power the turbine type rotation of a flagellum. And that flagellum gets rotating and rotating like a propeller, and it moves the cell along. So the PMF can also power the movement of a cell. So the PMF serves to power a lot of things. It is a very important aspect of the cell. And I want to make one more quick note on this. That is, prokaryotes capable of respiration or photosynthesis will have the components of the ETC across their membrane. If prokaryotes are not capable of respiration, that is, maybe they're strictly fermenters, they're not going to have the components of the ETC across their membrane. I want to look closer at the type of respiration that's being used by this particular bacterium. And by the way, this is the ETC of an E. coli cell that I have pictured here. Notice that the last acceptor of the electrons, I sometimes like to say that it's like an electron relay team. So the one that gets the baton last, or the electrons last, it's oxygen. So what kind of respiration is E. coli using in this figure? Now that's the last thing that you need to include on that extra credit that you emailed to me. Remember that I'm only going to accept that extra credit on Wednesday. I'm only going to accept it if it has answers to all 3 questions that I posed in this vodcast. And I'm hoping you're having a rocking Wednesday. See you soon.